Oo, alam ko naman ha. Pero naman siya para ano.
Oo, sige.
Pagkaan naman
Naintindihan naman natin yung activating factors ng tayo. Thank you, thank you.
Pero sa may sinabi ko kayo ng Maria. Kasi parang nakikita ko, gagamitin ko na ng mga bata yung mga dati. Parang nag-suggest ko yung parang nag-suggest ko, pwede bang surrender nila sa akin, sa mga advisors, yung 7-8 na nila may tapos babalikan mo pag closing na.
Match nga yan.
Sabi ko nga sa hindi ko lang bakit yung mga bata sa mga nag-agree na. Hindi ba yun?
Kasi pwede naman.
Diba? Surrender muna. O hindi na magamit na. Kasi ano sa kayo sabi naman ng kaya, sige po, dadali ko kami. Pwede ko kasi siya magagamit na lang ang realize na. Surrender na lang ako.
Tingnan muna natin. Mag-usapan muna ng guidance kayo kasi sa topic ng first. So, dapat nga coordinate ng first. Hindi naman ganun.
Yes. Thank you po. Lagi ko po lahat doon, ma'am, para isang announcement na lang ako. Di ba, guys? Yes, ma'am. Hi, everybody. Minsan gusto ko nung maging kabuti na lang. Gusto mo na, ma'am? Maging? Maging kabuti. Madami yung trabaho. May cash ka pa. May lahat pa rin. Marami lahat. So, wala na natin, hindi na siya Thursday. Wednesday na siya. Para meron tayo. Allowance. Available tayo. Then, may tatanong. Yes, sir. Uy, ganda ng mata. Isa mo lang. Ulit mo naman. Napili ka na naman. Order ka kayo. Is it like that? Sa may ano? Ano, makalilig. Kailan mo pasana ng papers? Pasok. Madali yan ang ano. Compute lang. Gusto mo?
Hindi naman po kasi nasa
Hindi
stage
ka
lang.
naman. Kasi yung bike naman.
Hindi, ako, yung isa ko talaga essential. So, hindi yung magsusunod ginawa ko. Madali lang kung lahat ng section na
Ang isa-send lang natin, ma'am, yung request sa data.
hindi mo, hindi nyo magagawa yun, hindi mo pwede work. Bakit, di ba ay, hindi. Kasi, kung isa ako di ba, isa ako. Pero na lang ako nabayad siya.
Good afternoon, class.
Pero kung ano, kasi, alam mo, ako lang na, alam mo, kilala ko lang ako. So, kung sa number 2 mahirap, so kung halos perfect, ano ko lang siya doon? Gawin mo lang po.
Kailan ako na nga ibang
Ganun lang. So, ano po siya? Para hindi niya, hindi siya alam mo. Ito yung mga tao, hindi siya. Wala siya.
kasi yan yung mga
Pero,
baka magtanong, bakit may isod wala akong request?
yes.
Kaya yung sa exam, nakalawa kasi mo kailangan ako.
Are the main sources of energy in the body, and brain cells and red blood cells are almost exclusively dependent on carbohydrates, that is glucose, as the energy source. These carbohydrates are widely distributed both in animal and plant tissues, and they are also called as saccharides. In Greek, saccharon means sugar. So carbohydrates are the substances containing carbon, hydrogen and oxygen, and they have general formula CnH2ON. Most of the common carbohydrates
Tapos, halimbawa sa akin nung sila ang aking mga nag-team.
Kunyari, ito ma, set, grade 10, ito, papers, yan na yun.
Oo. Tapos, halimbawa si, tawag ito, si paper sa, si, yan. Yung first one there.
Good afternoon, class. So, we are about to discuss carbohydrates today. So, please watch the video so that you will have a good idea of the role of carbohydrates in our body, the different types of carbohydrates, and then the functions of carbohydrates. Okay, so please watch the video. Please do take down notes on about those that I have mentioned. The function, the structure, and the different types of carbohydrates. Okay.
Carbohydrates confirm this formula, but some do not. And some carbohydrates also contain additional groups like nitrogen, phosphorus, or sulfur. So, what is the definition of carbohydrates? Carbohydrates are defined as polyhydroxyaldehydes or ketones or compounds which yield these on hydrolysis. What is meant by ketones? The compounds having this keto group. And what is meant by aldehydes? The compound having this aldehyde group. Carbohydrates are classified as monosaccharides, means simple sugar. Sugar
Babay ko, ma'am. Pumunta pa ako ulit.
Goodbye, ma'am.
Awan minute.
Bye-bye.
is soluble in water and sweet to taste. These monosaccharides, they are the simplest form of carbohydrates because they cannot be further hydrolyzed into simpler units. So this is the simplest form of carbohydrate. Disaccharides, they contain two monosaccharide units linked together by glycosidic linkage. Oligosaccharides contain 3 to 10 monosaccharide units joined together by glycosidic linkage. And polysaccharides, they contain more than 10 monosaccharide units. Now let's talk about each group one by one. So first is monosaccharides. Monosaccharides are simple sugar and they are the molecules having one actual or potential sugar group and they cannot be further hydrolyzed into smaller units. And these monosaccharides are further classified as aldoses and ketoses, and it depends on the presence of aldehyde group and ketone group. If the monosaccharide has aldehyde group, they are called as aldoses, and if they have keto group, then they are called as ketoses. Now let's see the various examples. Glyceraldehyde is aldose sugar and dihydroxyacetone is ketose sugar, and both glyceraldehyde and dihydroxyacetone, they contain 3 carbons, and that's why they are also called as trioses. Erythrose is aldose sugar. Erythrolose is ketose sugar and they contain 4 carbons, so they are called as heteroses. Ribose is aldose sugar. Ribulose is a ketose sugar and both contain 5 carbons, so both are pentoses. Glucose is aldose sugar and fructose is ketose, and both glucose and fructose, they contain 6 carbons and that's why they are hexoses. Glucoheptose is aldose. Pseudoheptulose is ketose and as heptoses. This is the structure of beta D glucose. It contains 6 carbon, and why it is called as beta D glucose? Beta means the hydroxyl group which is attached to carbon 1, it lies above the plane of ring and that's why it is called as beta glucose. And D means it is a D isomer. Most of the monosaccharides in the living beings, they belong to D series, and D glucose is the most abundant monosaccharide in the nature. And this is the structure of alpha D fructose. Why alpha? Because the hydroxyl group which is attached to the carbon 1, it lies below the plane of ring and that's why it is called as alpha. Disaccharide contains 2 monosaccharide units, and these 2 monosaccharide units, they are linked together by glycosidic bond. And disaccharides are crystalline and water soluble and sweet to taste, and they are further subclassified on the basis of presence or absence of free reducing group. So the disaccharides are classified as reducing disaccharide or non-reducing disaccharides. The reducing disaccharides examples are maltose and lactose. Maltose contains alpha D glucose plus alpha D glucose, means 2 molecules of alpha D glucose. They are linked together by alpha 1,4 glycosidic bond. And what is the constituent of lactose? 1 molecule of beta D galactose plus 1 beta D glucose, they are linked together by glycosidic bond to form lactose. So maltose and lactose, these are reducing disaccharides. What is the example of non-reducing disaccharide? It is sucrose. Sucrose is non-reducing disaccharide and it contains 1 molecule of alpha D glucose plus 1 molecule of beta D fructose, and both molecules, they are linked together by glycosidic bond. Oligosaccharides, they contain 3 to 10 monosaccharide units. They do not occur as free entities in living beings and they occur as glycoconjugates like glycoprotein, glycolipids. In plant world, the examples of oligosaccharides are raffinose, which contains 3 monosaccharide units like alpha D glucose, beta D galactose and beta D fructose. Stachyose is other example of oligosaccharide which contains 4 monosaccharide units, 2 galactose, 1 glucose and 1 fructose. Verbacose, it contains 5 monosaccharide units, 3 galactose, 1 glucose and 1 fructose. Raffinose are present in beans, cabbage, broccoli, etc. Stachyose are also present in vegetables and plants. Verbacose, they are present in legumes and they are fermented by intestinal bacteria and they cause flatulence. Coming to the polysaccharides, they contain more than 10 monosaccharide units. And these polysaccharides are also called as glycans or complex carbohydrates, and they have high molecular weight and they are sparingly soluble in water. And they are not sweet in taste and do not exhibit any properties of aldehyde or ketone group. And they are further classified as homopolysaccharides and heteropolysaccharides. Homopolysaccharides contain same type of monosaccharide units, and heteropolysaccharides contain more than or equal to two different types of monosaccharide units or their derivatives. So polysaccharides are of two types, homopolysaccharides and heteropolysaccharides. Homopolysaccharides are also called as homoglycans and heteropolysaccharides are also called as heteroglycans. The examples of homopolysaccharides are starch, dextrine, glycogen, cellulose, inulin, dextran, chitin. And the examples of heteropolysaccharides are agar and agarose, gum and pectin, and various GAGs like glycosaminoglycans like hyaluronic acid, chondroitin sulfate, keratin sulfate, dermatan sulfate, heparan sulfate. So these are called as glycosaminoglycans or GAGs, and earlier they were called as mucopolysaccharides.
Don't skip Nido 3 Plus to help protect your toddler against coughs and colds.
Now let's enumerate the functions of carbohydrates. They are the most abundant dietary source of energy. 4 kilocalories are formed when 1 gram of carbohydrate is oxidized. They are the storage form of energy in the form of glycogen. Glycogen is stored in the liver and muscle. The non-digestible carbohydrates like cellulose, agar, gum, and pectin, they are dietary fibers. They are the precursor of organic compounds. For example, ribose and deoxyribose, they are the important constituents of nucleic acids like DNA, RNA, and they are precursor of compounds like glycolipids, glycoproteins, and proteoglycans. Carbohydrates participate in structure of cell membrane because glycolipids and glycoproteins are the constituent of cell membrane. They play a role in lubrication because the GAGs like hyaluronic acid is important for lubrication of joints, cellular intercommunication, and immunity. They serve as structural component. For example, glycosaminoglycans in humans, cellulose in plants, and chitin in insects. And they are involved in detoxification reaction. For example, derivative of carbohydrates, glucuronic acid is involved in detoxification process. So let's now summarize the topic. Carbohydrates are defined as polyhydroxyaldehydes or ketones or compounds which yield these on hydrolysis, and they are classified as monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Monosaccharides are simple sugar. Disaccharides consist of 2 monosaccharide units. Oligosaccharides consist of 3 to 10 monosaccharide units. Polysaccharide consists of more than 10 monosaccharide units. The example of monosaccharides are aldoses and ketoses. Glyceraldehyde, erythrose, ribose, glucose, glucoheptose, these are aldoses, while dihydroxyacetone, erythrulose, ribulose, fructose, and pseudoheptulose, these are ketoses. The examples of disaccharides are the reducing disaccharides and non-reducing disaccharides. Maltose and lactose are reducing disaccharides, while sucrose is non-reducing disaccharide. The example of oligosaccharides are raffinose, stachyose, verbacose. And polysaccharides are further subdivided into homopolysaccharides and heteropolysaccharides. Starch, dextrine, glycogen, cellulose, inulin, dextran, chitin, these are homopolysaccharides. And agar, agarose, gum and pectin, hyaluronic acid, chondroitin sulfate, keratin sulfate, dermatan sulfate, heparan sulfate, these are heteropolysaccharides. glycerine, glycogen, cellulose, inulin, dextrose.
Okay, class, can you hear me? Am I audible?
Audible po, pero may echo lang po.
Ah, okay, okay. Wait lang ha. May diferensya yung aking mouse. Wait lang.
Kaya na muna sir. Ito na ako sa kabila.
Oo, sige.
Yung huli babae.
Sige, babay po.
Mag-i-shoot na tayo.
Oh, sige, sir. For a while, class, ha, nawala lang yung connection ko doon sa isa. Sandali lang. Okay, so you have heard about the carbohydrates. First, you have learned about the different types of carbohydrates. Second, we have also heard the different functions of carbohydrates. Okay, can you hear me now, class?
Yes po.
Okay. So, uh, we have learned also that carbohydrate is the most abundant of all the um, biomolecules. Okay, so and it is composed of carbon, hydrogen, and oxygen. Now, so with the structure, we will be able to um, predict what chemical reaction these carbohydrates will undergo. Okay, so after knowing, okay, before I proceed with our discussion, let me ask you again, what are the three types of carbohydrate based on the number of sugars, on the number of sugar present in it? What are the three types of carbohydrates? You may use the chat box if you want. What are the three types of carbohydrates? Class? Okay, thank you, Isabella. So, those are the three types of carbohydrates. Okay, so uh, what type of bond is present in disaccharides, oligosaccharides, and polysaccharides that connects to monomers of carbohydrates? What do you call that type of bond?
You have the what do you call that type of bond? Yes, you have the glycosidic bond. Now, so with this, there is a for our polysaccharides, we have an alpha polysaccharides and a beta glycosidic bonds. Okay, so you have seen from the video the difference between an alpha and a beta glycosidic bonds. And the structure of the carbohydrates is very important for the chemical reaction that it will undergo. Okay, let us look into the chemical reaction that our carbohydrates undergo. Nalilito.
Qualitative analysis of carbohydrates. Carbohydrates are large biological molecules consisting of carbon, hydrogen, and oxygen atoms having the general formula of CmH2O n times. Carbohydrates are one of the important nutrients present in our food. During the process of photosynthesis, carbohydrates are produced from carbon dioxide and water in the presence of chlorophyll and sunlight. Glucose, lactose, sucrose, and starch are commonly known carbohydrates. Our aim here is to learn how to conduct a few simple tests to analyze the properties and reactions of 4 common carbohydrates: glucose, lactose, sucrose, and starch. The tests are solubility test, Molish's test, Feeling's test, Benedict's test, Tollens test, iodine test. Materials required: glucose, lactose, sucrose, starch, distilled water, aqueous solutions of glucose, lactose, sucrose, suspension of starch, Molish's reagent, concentrated sulfuric acid, Feeling solution A, Feeling solution B, Benedict's reagent, Tollens reagent, iodine solution, test tubes, droppers, and water bath. Solubility test. Take small amounts of glucose, lactose, sucrose, and starch in 4 test tubes A, B, C, and D respectively. Now, add a small volume of distilled water to the test tubes containing glucose, lactose, sucrose, and starch. Shake each test tube well. We can see that glucose, lactose, and sucrose are soluble in water, whereas starch is insoluble in water. Molish's test. Take a small quantity of aqueous solutions of glucose, lactose, sucrose, and suspension of starch in 4 test tubes A, B, C, and D respectively. Using a dropper, add a few drops of Molish's reagent to the test tubes A, B, C, and D. Take the test tube A and pour a small amount of concentrated sulfuric acid slowly along the side of the test tube. Similarly, pour concentrated sulfuric acid to the other test tubes B, C, and D. Concentrated sulfuric acid dehydrates carbohydrates, to form furforaldehyde or its derivative, which further reacts with alpha naphthol present in the molicious reagent to form a colored product that appears as a purple ring at the interface between the acid layer and the test layer. Feelings test. Take a small quantity of aqueous solution of glucose, lactose, sucrose and suspension of starch in 4 test tubes A, B, C and D respectively. Using a dropper, add a small quantity of feeling solution A into test tubes A, B, C and D. Now, using another dropper, add a small quantity of feeling solution B into the test tubes A, B, C and D. Heat the test tubes in a boiling water bath for some time. The reducing sugars, glucose and lactose, reduce the copper 2 ions in the test reagent to form red precipitate of cuprous oxide, whereas no such precipitate is formed from the non-reducing sugars, sucrose and starch. Benedict's test. Take a small quantity of aqueous solutions of glucose, lactose, sucrose and suspension of starch in 4 test tubes A, B, C and D respectively. Using a dropper, add a small quantity of Benedict's reagent into the test tubes A, B, C and D. Heat the test tubes in a boiling water bath for some time. The reducing sugars, glucose and lactose reduce the copper 2 ions in the test reagent to form red precipitate of cuprous oxide, whereas no such precipitate is formed from the non-reducing sugars, sucrose and starch. Tollens test. Take a small quantity of aqueous solutions of glucose, lactose, sucrose and suspension of starch in 4 test tubes A, B, C and D respectively. Using a dropper, add a small quantity of Tollens reagent into the test tubes A, B, C and D. Heat the test tubes in a boiling water bath for some time. The reducing sugars, glucose and lactose reduce silver ions in the test reagent to elemental silver, appearing as a silver mirror on the inner surface of the vessel. No such silver mirror is produced by non-reducing sugars, sucrose and starch. Iodine test. Take a small quantity of aqueous solutions of glucose, lactose, sucrose and suspension of starch in 4 test tubes A, B, C and D respectively. Using a dropper, add a few drops of iodine solution into the test tubes A, B, C and D. Iodine reacts with starch to form a blue-colored starch-iodine complex, whereas no such complex is formed by other samples.
Okay, so we have performed this general test for carbohydrates. So basically, the first test is the Molish test. And in the Molish test, that is a general test for carbohydrates. Okay, so we have seen that in the Molish test class, the carbohydrates, the carbohydrates class, it reacts with the Molish reagent which contains alpha-naphtol in ethanol. Okay, please remember that our Molish reagent contains alpha-naphtol in ethanol. And that naphthol form a purple or purplish red ring at the point of contact between our sulfuric acid and the analyte. Our analyte, of course, is our sugar, and with our reagent, the Molish reagent. And we will be able to see class in the Molish test that purple, the red ring. Oh, okay. So there are some sugar that that do not react with Molish test. What are those kind of sugar? Those are the tetroses and the trioses class. So if we try to test our tetroses and trioses, like our glyceraldehyde and dihydroxyacetone, an example of an aldose and a ketose, which is which has 3 carbon, it will not react with our Molish test. Okay, so it is not capable of uh, undergo reacting with sulfuric acid because they easily undergo hydrolysis when exposed to strong mineral acid. Same with glycoprotein and okay, the same with glycoprotein. So, what happens during the Molish test? The carbohydrate undergoes dehydration, which means that the water, upon the introduction of our sulfuric acid, is released and result in the formation of an aldehyde, which undergoes condensation with as it reacts with our naphtol. Ayun. So, yun yung nangyayari doon, class. So, there is a hydrolysis reaction. There is a dehydration reaction upon the addition of sulfuric acid. So, when we say dehydration, class, it means that the sulfuric acid is removed from your sugar. Okay? So, exceptions are the tetroses and trioses. Now, the second reaction that we have for our sulfuric acid class is the reaction with Felling's reagent. Okay? So you can see that upon heating your sugar, if it is reduced, it will what? It will produce a red precipitate. So in your test substance, which among your sample reacted with the Felling's reagent? With ano yung nag-react sa inyo? Sa inyong uh, sample. Okay, let's check the different samples that we have for this experiment. Ano ba yung mga sample natin? Do you still remember? Naalala niyo ba yung tinest natin, class? Ala, nakalimutan niyo na. Ano ang ating mga tinest sa Felling's test, class? Ano yung tinest natin? So, for the Felling's test, we have xylose, sucrose, and starch. Tama ba? Do you still remember that one? So our test samples are xylose, sucrose, and starch for the Fellings reagent, for the Fellings test. Sige, wait lang anak. Wait lang. Do you have your data sheet with you? Sandali. So, doon sa tatlo, xylose, sucrose, and starch, alin ang mga nag-react with our Fellings reagent? Which gives a red precipitate in our Fellings test. Do you have your data sheets with you? Class? Okay, based dun sa video na pinanood natin, which do you think is not capable of reducing sugar? Reducing copper. Sucrose. Okay. Thank you, Isabella. So sucrose cannot be a reducing agent. Okay. So we cannot form copper 2 plus. We cannot form copper plus 1 when our sucrose is present. So that's why we should have a negative result in the sucrose analyte for the Fellings test. Okay? So Fellings solution, Fellings test is a test for reducing sugar because it converts our sugar, this RCHO, that is our sugar, and that will be converted into an acid. Okay? And while your copper 2 plus will become copper plus 1. Okay. Your sugar is oxidized while your copper is reduced in the Fellings test. Another test for the ability of our sugar to reduce, to become a reducing agent is the Benedict's test, di ba? We perform the Benedict's test and also the Nylander test. So, for the Benedict's test, it gives a positive result for fructose, glucose, maltose, and lactose. And again, it will give a negative result for our sugar because our sugar is a non-reducing sugar. So, what is the color change in our Benedict's test? We will also produce a blue to brick red precipitate. Okay? After heating our sample in a water bath. So, Again, Benedict's reagent contains blue copper. 2 is reduced to copper 1 and precipitated as copper oxide. So, for our experiment, our sugar again that was tested using our Benedict's reagents are xylose, sucrose, and starch and glucose. And again, it gives a negative result for your sucrose. Did you get that one from your, uh, did you observe that one? Class? Na-observe nyo ba yon? Were you able to observe? Salamat si Isabella, ang aking estudyante ngayon. Okay, sige. Then another reducing test is the Nylander's test. So for this one, we use naman, our chemical is bismuth subnitrate. So the positive result for this one is the formation of a black precipitate. Okay, so do we have a Nylander solution? I forgot. Nakalimutan ko. Meron. Okay. So, medyo nakalimutan ko ito eh. So again, it will only be sucrose that will not react with our reducing agent. Okay, class? So bear in mind that this one, for all these three tests, Benedict's test, Nylander's test, and also our Felin's test, they are all a test of the reducing ability of our sugar. Okay. In the process of reducing the metal ions like copper or bismuth, your sugar in turn is changed into a carboxylic acid because they are oxidized. So, the next test is I think it's the Moore's test. Our next test now will be the Moore's test. Wait lang ha. Okay, so I think, did we perform the Moore's test class? Nakalimutan ko. So, what are the reagents for our Moore's reagents? So our reagents will be barium hydroxide, sodium hydroxide, and barium hydroxide. Okay. So, for this one, our our analyte is only glucose and sucrose. Okay. It is also a reducing test. That's why in this case, your sucrose will not give any positive result for your Moore's test. Okay? So, in the Moore's test, the positive result is the formation of your uh, caramel, the formation of a caramelized structure or the browning of our sugar. Okay? So, we all know, class, that this Moore's test is also a reducing test for sugar. So, again, our barium will become, the barium will be precipitated and the sugar will be caramelized and the brown color is the positive result for the Moore's test. So those are general tests for carbohydrates. So basically, we have two tests for this. Our carbohydrates undergo two chemical reaction. One is dehydration and the other is redox reaction wherein our sugar act as a reducing agent and it is oxidized into what? It is oxidized into a carboxylic acid. Okay? So in the process, in the reducing agent test, we conducted 4 tests in the lab. We have the Felling's test, the Nylander's test, the Benedict's test, and the Moore's test. Okay. So those 4 tests are tests for reducing sugar. And in that 4 test class, sucrose gives a negative result because it is a non-reducing sugar. Okay? Maliwanag na po ba yun sa atin? Yung the test that we perform, okay? So, you know the color change for Felling's test and Benedict's test. We form a red colored precipitate. For bismuth, it is a black color. And for the Moore's test, we have what? We have a uh, we have a caramel colored substance. Okay. Aside from that, let's continue. Our carbohydrates undergo hydrolysis. Now, what is the meaning of hydrolysis? So hydrolysis is the process of using water to break a bond. Okay, so when a bond is broken, what happens is there is a release of energy. So what usually are the type of carbohydrates that undergo hydrolysis? Usually those are your starch, glycogen, and dextrine. They are the polysaccharide. Okay? So now when our polysaccharides hydrolyzes, it gives a positive result with the iodine test. So if we are to determine sugar is whether a sugar is undergoing hydrolysis, that is the use of water to break a bond, okay, we will be using the iodine test, okay. So when we subject our starch to iodine test, you know that the positive result already is turning from iodine color from orange or brownish color to black or blue. Okay? So, what is the meaning when our iodine solution is turned into a blue colored solution? It means that the bond in our polysaccharide, especially starch, is broken. Evidently, we have seen this in our previous experiment when our macaroni class is submerged into into pancreatin and salivary amylase and sodium carbonate. You have seen that all of those chemicals are very strong hydrolyzing agent that breaks the bond in our starch present in our carbohydrates. Okay? So that is the hydrolysis. So again, hydrolysis reaction means the use of water to break a bond. And this hydrolysis is happening in our polysaccharide like starch with the presence of the enzyme and other agents that will induce breaking of the bond between polysaccharide. Okay, so the next test class is, so let's watch this video on hydrolysis. At the end of video, we will answer this one already.
Hydrolysis.
What are the two types of starch? What is the monomer of starch? And draw the structure of the monomer. Okay? So, we will answer this one today after watching the video. Hindi ko na ipapadraw yung structure class. Okay, just give me the types of starch and the monomer of starch. Make sure that in the lab, you should be able to draw the monomer, which is your glucose molecule.
This is the process of adding water to a polysaccharide molecule in order to break it down into smaller units. Remember, we can take simple sugars, or monosaccharides, and join two of these together to form double sugars, or disaccharides. This process of forming disaccharides from monosaccharides is dehydration synthesis. Many disaccharide or monosaccharide units can also be joined together to form polysaccharides, also by the process of dehydration synthesis. Now we can also break down polysaccharides into disaccharides, and this is done using the process of hydrolysis, or adding water. Disaccharides and polysaccharides can also be broken down into monosaccharides, again using the process of hydrolysis.
Okay, class. So as you can see from this video, dehydration, if you remove water, you can join two monosaccharides together. Okay, and further reduction of water can result into a polysaccharide. When we say reduction of water, why? Because that is dehydration. Okay, so now, if water is added and is used, water now can be used to break down the bonds between a polysaccharide and use it to produce disaccharide. Disaccharide through the process of hydrolysis. That means the adding of water, the use of water to break the bonds present in our uh, disaccharide to produce a monosaccharide. Therefore, water can be used to break down and convert our disaccharide into monosaccharide. Okay? So dito, removal of water sa dehydration. Dito, the use of water in order to break the bonds in our carbohydrates. So this is the monomer class. This is the monomer, the structure of our glucose. When our disaccharide and polysaccharide are broken into its uh, simple sugar. Okay? So this is glucose.
To summarize, dehydration synthesis is used to build larger molecules from smaller ones, and hydrolysis is used to break down larger molecules into smaller ones. Let's take a molecule of the polysaccharide starch, and we'll focus on the first part of the chain consisting of 4 subunits. With the appropriate enzyme present, we can add a molecule of water. And using the process of hydrolysis, the starch molecule breaks apart and water is added. And now a maltose molecule has broken off of the large starch molecule. This breakdown of starch into maltose using hydrolysis takes place in the mouth with the help of the enzyme salivary amylase. Now we'll consider this molecule.
Okay. So, initial digestion of our starch is taking place in our mouth. Okay, with the help of the salivary amylase. So, you have seen that starch breaks down into maltose. Okay, with the help, of course, of your salivary amylase.
Molecule of maltose. With the proper enzyme present, maltose can also undergo hydrolysis. And as water is added, break up into 2 molecules of glucose. The breakdown of maltose to simple glucose molecules takes place in the small intestine, as well as breaking down.
Okay, wait lang class ha. So please study the structure of our glucose molecule. So this is the ring formed of our glucose molecule, which is a 6 this is a 6 carbon molecule of carbohydrate. So you will see this is carbon 1, 2, 3, 4, 5, 6. 1, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and this one is your carbon 6 class. Okay? For a glucose molecule. And the breaking down of your maltose to glucose molecule is taking place in our what? In our small intestine.
Maltose to simple glucose molecules takes place in the small intestine. As well as breaking down starch, the process of hydrolysis is also used to break glycogen down into glucose when a quick energy source is needed for muscles. But remember, our body does not possess the enzymes necessary to break cellulose down into glucose.
Okay, class. By the way, glycogen is the stored polysaccharide of human and animals, while your starch is the stored polysaccharide of your plants. Now, let us answer the question in our for this video. Okay, so what are the two types of starch? I think what is meant here is what are the two types of polysaccharide mentioned in the video? Actually, meron naman talagang two types of starch. Sige, what are the two types of starch? But I did not hear it in the video. Do you know what are the two types of starch, class? Was it mentioned in the lecture class? The two types of starch is amylose and amylopectin. Okay, yes. Tiffany, you're right. Those are the two type of starch, amylose and amylopectin. Now, what is the monomer of our carbohydrates? What is the monomer of our starch? Let me see. What is the monomer of our starch? You have seen from the video? Okay, yes, you're right. Glucose is, yes, you're right. You're right, Eliza. You're right, Tiffany. That the monomer of our starch is glucose. And you have seen the ring structure of glucose. Okay? Just remember, di ko na to papadrawing. Okay? Sa inyo ngayon. But let us look back into the structure of glucose. Pagbibilangin ko uli. Glucose is a 6-carbon ring. Okay? This is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. And your carbon 6 is projected upward. And it contains CH2OH. Okay. Now, 1, 2, 3, 4, 5, 6. Okay. So that is the structure of our glucose molecule. Okay? So, if your carbon, if that if we will have the Fisher projection of that carbon class, wait lang, the Fisher projection class, paano yung Fisher projection ng ating carbon? Sandali, ayaw gumagano. Okay. So this will be the Fisher projection of our carbon. So this is 1, 2, 3, 4, 5, 6. Okay? So this one will be the carbon that is projected upward in the ring structure. This is the fifth carbon. Okay? So this is how our carbon will look like in the Fisher projection. Okay? So, let's go back to our class. To our topic pala, to our class. Wait lang anak. One, two, three. So, this will be on the left, on the left, on the right, on the left. Okay. One, two. Okay, so let's continue with our test. Now we will be studying the special test that our carbohydrates will undergo. And what are the three special tests that we conducted for our carbohydrates class? We have the Bayer's Arsenal test and also, wait, I don't have internet. Sandali lang, anak ha, nawawala yung internet ko dun sa isa kong device. Kaya pala. Sandali lang, class. Wala na naman ako. Sandali lang. Class, sandali lang ha. Ayusin ko lang ng internet. Sandali lang. Sandali. Hi, ma'am. Sige, ma'am. Ha? Sige, ma'am.
Hindi naman na ano ni sir. Ako na lang yung na-type niya. Dito ka na lang sa kabila.
Sige, sige. Bakit dalawa yung
Isa
ni...
lang. Every isa lang.
Isa lang. Sige, ma'am. Sa'yo muna. Ikaw kasi ang nakapagpirma. Oo, kasi hindi ka pa yung okay. Okay, so let's continue, class. So, the next thing that we will be discussing will be the special test for carbohydrates, okay? So, and what are those special tests that we perform for our carbohydrates? The Biel Arsenal test, the Seliwanov test, and I think we were not able to do the last test. Tama ba? Hindi natin nagawa yung last test, class. We did the test for complex carbohydrates. Actually, the iodine test. So, of course, you will notice that for the iodine test, let's look back to our iodine test first. Okay. So, we use 1% glycogen, 1% glucose, and also 1%, wait lang anak, starch. Okay. Which among the three did not react with the iodine test? Alin dito sa tatlong ito ang hindi nag-react sa iodine test, class? Which among the three samples? Glycogen, glucose, and starch. Of course, we know that starch will react with our iodine test. Alin yung mga nag-react? Did you see your glycogen and glucose reacting in the iodine test? Class, did you observe your glycogen reacting? I think yung iodine po, glucose and glycogen. Ayan. Thank you uli, Isabella. So, it is glucose and glycogen which did not react with your iodine. Okay? So, that is for that part. So, we know that the iodine test is a test for complex carbohydrates. Now, how about our Seliwanov's test? Okay. What happened to our solution based on the Seliwanov's test? So, what are our analyte here? We have fructose, sucrose, and also starch solution. Fructose, sucrose, and starch solution. What again is Seliwanov test? This is a test for what? Sinulat ko sa board. Ano yung tinitest sa Seliwanov test, class? What is being tested? What is the aim of the Seliwanov's test? Huh? It is a test for ketosis. So, okay. So, and the positive results here is the formation of a red color solution. Diba? And there is one. Which one will react in the Seliwanov's test? O, tingnan natin. Ano ba test reagent natin ulit sa Seliwanov's or analyte natin? We have starch, sucrose, fructose, and glucose. Okay. Alin dyan ang ketosis? There are two. Fructose and sucrose. Very good. Very good. So your fructose and sucrose are ketoses. Of course, sucrose, as we all know, is made up of fructose and glucose. And fructose is our table sugar. Okay. So fructose is a 6-carbon sugar also that is a ketose. So let us look into the structure of our fructose. Okay. Wait lang, anak. Ayan. Okay. So this is our fructose class. So you see that the second carbon, you have the carbonyl group on the second carbon. Okay. Now, so we have the D and the L fructose now. Seemingly, when it is formed into a ring, what you have there, class, is a 5-carbon ring. Okay. It's a 5-carbon ring, but fructose is a 6-carbon molecule, even though if it is formed in a 5-carbon ring. So, ito yung fructose class, ha. So, this is our double bond O, and it is attached to two carbons. Okay, unlike our glucose, it is an aldose. Okay, C double bond O H. Diba? So, this is our fructose. Tandaan, ha, kung nasaan ang double bond, nasa second. Now, we said that if this is fructose, let us look into the structure of our sucrose. Okay. So, this is the structure of our sucrose class. So you can easily see that this is the fructose and this one is your glucose molecule. Okay? So that's the structural formula of our sucrose. So you have here a 5 carbon and a 5 ring. This one is a 6 ring sugar. This one is a 5 ring sugar. Okay? That's why, and we know that the 5 ring is the fructose, and fructose is a ketose. Therefore, it will give a positive result in the Seliwanov's test. Now, the last one is our Bial orsinol test. What is being tested in Bial orsinol? Bial orsinol test. Okay. What is being tested in our Bial orsinols test class? Pentoses. Okay, so now, so what are our, tama, very good. Thank you, Isabella. In the BL orsinols test, we have fructose, xylose, and glucose. So, which one reacted with BL orsinol to form a blue-green? Blue-green, tama ba? Blue-green solution. Oh, Xylose. Okay, so thank you, Isabella. It is xylose that reacted. Kasi you have seen from the previous structure that fructose is a 6-carbon sugar, while xylose is a 5-carbon sugar. Let us look into the structure of your xylose, okay? Okay, class. So, so this is the structure of our xylose. 1, 2, 3, 4, 5. It's a 5-carbon sugar, which is a pentose. So that is the information, okay, of your xylose. The ring and the chair conformation, the Howard projection, and if you will notice, class, look here. 1, 2, 3, 4, 5. You have the 5 carbon. There, on the 5th carbon, there is no more. It ends at the 5th carbon. So it is a 6-ring sugar. Tama, 6-ring to eh. But it is a 5-carbon sugar. And this is the Fisher projection of our xylose. So, madi-differentiate na natin, di ba? Okay, alam natin, pagka ito ay xylose. Pagka yan ay glucose, may another CH2OH pa dito. Okay? So, that's all that we did for last meeting. Okay? So, you have been familiar with the structure of xylose, glucose, sucrose, and fructose. And we also discussed the different tests like Beal's orsinol test for pentoses, our what else? Our Seliwanov's test, the test for ketosis, and the complex carbohydrate test, which is the iodine test. Then we also what? We also studied the different the general characteristics of sugar, that they are able to form polysaccharides through the process of dehydration, and the breaking down of the polysaccharides takes place to the process of what? What you call the process of breaking down, it is hydrolysis. Okay, class. Now, we also learned about the reaction that our carbohydrates undergo and its solubility. We also learned in the video the function of our carbohydrates. Okay, I will just discuss first a little of lipids. Okay, para for those who will not be present in our lecture on Monday, but this will be an on-site lecture, you'll have an idea of what we will be discussing. And also on Monday, uh, there will be a worksheet activity for those who will be present. Okay, and that will serve as a reviewer for our midterm examination also. Okay, so let's talk about lipids. Tama ba? End na ba yun? Wait lang, anak, titingnan ko kung end na to ng carbohydrates. Remember, class, that carbohydrates is the source of energy of all living organisms, because our carbohydrates, glucose, produces the energy, adenosine, the source of energy in our body, which is ATP, or adenosine triphosphate. Now, for some, glucose is, or carbohydrate serves for structural support, okay, like the chitin of of insects and some sea organism. It's a kind of complex carbohydrates that serves as an exoskeleton for insects and other sea organisms. Now, cellulose is another structural support class for our plants. Okay, so sometimes together with the protein, our carbohydrates serve in the in the cell membrane. So carbohydrate, it is also detoxicant. Okay, it detox our body, especially the fibers. We all know that the fibers coming from the plants are the cleaners of our body. Okay, so those are the importance of carbohydrates that we have seen in the video. So our next topic, class, is lipids. So lipids, class, are what? We sometimes refer lipids as these are fat molecules, okay? So these lipids are very big micromolecules. Similar to carbohydrates, it is also made up of carbon, hydrogen, and oxygen. Now, similar also to carbohydrates, they are related to energy. However, lipids are the stored energy in our body. So the monomer of lipids are fatty acids. Okay. So fatty acids and glycerol. So most lipids are insoluble in water because they are non-polar. So you know that if they are non-polar, they have no positive or negative charge. Unlike carbohydrates, again, I will always refer to carbohydrates because small molecules of carbohydrates are soluble in water. However, big or polymers of carbohydrates, our polysaccharides like cellulose, some are not soluble in water. Now, as you can see from, so let's go back to our lipids, which is a non-polar molecule. Okay, you know that lipids cannot mix with water. Okay, they are not the same. There is a principle in solubility that like dissolves like. Since water is a polar molecule and lipid is a nonpolar molecule, therefore lipid will not dissolve in water. Moreover, uh, lipids are what? They contains a polar head and a non-polar tail. Okay? So this is your polar head and inside is the non-polar tail. Okay, now you will see that the polar head, though it's hydrophilic, it is not dissolved in water pa rin. Okay, now this illustration is a representative illustration. Ay, hindi ko pala ito na-share. Share the tab instead. Kanina pa ako imik ng imik. Sorry, class, ha? So, that is the formula of, but not the formula, the structure of the lipid layer in our cell membrane. So, our cell membrane is a lipid bilayer. Why is it a lipid bilayer? Because there is a hydrophilic head and a hydrophobic tail. Okay? So, that now, as I mentioned, lipid does not dissolve in water. And okay, now, so, wait lang, anak. So, let us look into the classification of lipids based on its structure. Wait lang anak, nawala ang aking class.
Group of hydrophobic biomolecules that play important roles in living organisms. While the primary function of lipids is long-term energy storage, lipids are also used for protection, insulation, and lubrication. They also act as precursors for some hormones and are a key component of cell membranes. There are 4 basic groups of lipids. These are triglycerides, phospholipids, steroids, and waxes. Although these groups differ in many respects, they all have one characteristic in common. They are all insoluble in water. You may have noticed that lipids and water do not mix. For example, notice the yellow-colored oil in the beaker of water shown here? Even if we stirred this for several minutes, or even several hours, the oil would still separate out from the water. This is because lipids are hydrophobic. From Latin, the prefix hydro means water, and phobic means fear of. So when you hear that lipids are hydrophobic, this basically means that water and lipids do not mix. Let's take a closer look at the category of lipids known as triglycerides. Triglycerides include the fats and oils. Fats, such as lard and butter, are solid at room temperature and are used by animals for insulation, protection, and long-term energy storage. Oils, such as corn oil and olive oil, are liquid at room temperature and are used by plants for long-term energy storage. At the molecular level, triglycerides contain 2 types of subunit molecules, glycerol and fatty acids. Let's take a quick look at fatty acids. A fatty acid has 3 main parts: a chain of carbon and hydrogen atoms called the hydrocarbon chain, a methyl group at one end, and an acid group at the other end. Fatty acids can either be saturated or unsaturated. A fatty acid that has only single carbon-to-carbon bonds is known as a saturated fatty acid. This is because the carbon chain is saturated with all the hydrogen atoms it can hold. Unsaturated fatty acids have one to several double bonds. Double bonds result in kinks in the fatty